Here's an example dataset of the spatial distribution of the value of PM2.5 in Baltimore a few years ago. Here, each data point denotes one sensor or PM2.5 device. And for each location, the PM2.5 values is provided, and the position of this sensor is also provided. Another example data is on the income distribution of different districts in Paris. Here, each data point is one district, and for each data point, the location of its district, as well as its median income value and tax of the people that living in that district is provided. Another example is the spatial data in the biological studies. Here is the spatial gene expression of this gene. Here, each spots is the locate. Here, each spot is a location that we are sequencing the gene expression. For each spot, the locations as well as the gene expression measures of each gene out of tens of thousands genes are provided. As we just see, spatial data is widely used a variety of disciplines. So what's special of this type of data? Firstly, this data provides us extra information on the data point level or observation level. Using the spatial locations, we could derive the spatial distance between different data points. And in using the distance, we can furtherly define the neighborhood for each data point. For example, here for any given district, we can use the distance information to get a list of neighboring districts. On the feature level or measurement level, such as the spatial distribution of PM2.5, the spatial data is allowing us to quantify the spatial dependency. Intuitive understanding of this terminology is the variance of the ratio of the variance that can be explained by the spatial locations and the spatial distances. An intuitive way to quantify this is to firstly to compare the variance within the neighborhood and the variance across different neighborhoods. And I will go through these definitions in details in the following slides. For two feature comparison, such as comparing the carbon dioxide spatial distribution with the PM2.5 value distribution, this type of data is allowing us to quantify the spatial correlations between these two features. Or in other words, see whether these two features are changing similarly across different neighbors. There are further characterizations of spatial data we could study. Firstly, we can define spatial domains based on the similarity of the data point while accounting for the spatial locations of this data point. For example, using the income data of different districts of Paris, we could partition the areas of this country into a high income part and a low income area. Furthermore, for a given feature, we could extract the spatial dependent part of this feature. And from a visualization point of view, we could do a two-dimensional smoothness of the feature, which will give us a smoothed surface that will better illustrate the spatial trend of this feature. There are many questions that can be answered by analyzing the spatial data. For example, does the PM2.5 distribution in Baltimore have a spatial pattern? If there is, can we identify a certain region or area that has more severe air pollutions compared with the surrounding areas, and which could furtherly inform the governments to bring more attentions in their policy makings. Secondly, we can ask whether the income level is spatially correlated with the housing rates in Paris. So far, we have described the data format and have a basic sense of the questions we could ask and solved by the data. Any questions so far? Okay. Next, I will introduce a few analysis tools for visualize and analyzing the spatial data. Firstly, I will introduce some visualization tools. For spatial data visualization, a natural way is to firstly plot the data points in a two-dimensional space using their spatial locations information. And for each point, we can color them by the value of a certain feature. However, if we want to further emphasize the spatial trends of this feature across the space, we could use a package called MBA, which could do a two-dimensional smoothing of the variable, and then using fills, which could create a color legend that shows the continuous color scale in the plot. And here is the functions of making the plots. And using these functions, we could create plots that more emphasizing the spatial trend. As we can see that after in the smooth plots, some local variations, like this area is smoothed out by the two-dimensional smoothing and creates, in this way, it creates a smooth two-dimensional surface. So far, we have went through the visualization tools. Next, I will move on to the analysis tools. As we just talked about, we can use spatial data to find out which features are spatial dependent by quantifying the spatial dependency of the data. In a statistical terms, the spatial dependency means that the value of a certain feature in a given data point is correlated with its value in the other data points, where such correlation is dependent on their distance. In another way, we can understand it as how much spatial how much the variance of this feature could be explained by the spatial locations and the spatial and the correlation structures. Here is the real distribution of the income, and here is a random distribution generated by simulation. And we can see that there is an obvious spatial trend in the real distribution, and you don't see a strong trend, spatial trend in the simulated data. Here are a few additional plots for the simulated data, where the spatial dependencies are increasing from left to right. By simply visualizing those spatial plots, we could tell that the plot on the right, the features on the right has more spatial patterns, has a stronger spatial dependencies compared with the features on the left. Next, we may ask how to better quantify the spatial dependencies. As we discussed in a few slides earlier, The spatial dependencies can be understood by the similarity of the relative variance of this of certain feature that can be explained by the spatial locations. And a feature with high spatial dependency would show close values between the adjacent data point or within a neighbor compared with the difference of the values with random selected or a far away data point. So this type of measure is transforming the spatial dependency problem into a marginal correlation problem by plotting the feature's value against the value of its neighbors. Here, for each dot, the x-axis is the income in one district, and the y-axis is the income in its neighboring district. And we can see that in the real data, there is strong relationship between the districts and its neighbors' income, while in the random data, we don't see any relationship. Doing the same plot for the other three sets of simulated data, we can also see a stronger linear relationship for the data sets with strong spatial dependencies. Okay, next move on to an example data. This data is about the ozone concentrations on the Los Angeles country. Here the colors is denoting the ozone concentration level. Here I'm introducing a package called APE to quantify the spatial dependency of the data. And the data here can be downloaded from the UCLA website. Here is what the data look like. This latitude and the longitude information denoting the locations as well as the ozone concentrations. This package is using the similar idea of what I have just described a few slides ago, where the scatter plot was made for marginal correlation comparisons. And here I don't have time to go through the details, but using this method, we can have a p-value that can tell us how significant the spatial dependency is for this certain variable compared with a randomly distributed variable. Here I'm listing a few packages that could be used to calculate the spatial dependencies. There are different assumptions under different packages. As we just talked about, the spatial dependencies is defined by there's a spatial the correlation between the spots is a function of the distance of the spots, and how the correlation would be. The certain assumption would be made, such as a linear relationship or an exponential relationship. And different assumptions could lead to different likelihood and modeling, and there are different packages that uses different assumptions. So far, we have talked about how to find out features that are spatial dependent. Next, let's move on to quantify the spatial correlationships between a pair of features. In other words, how to measure the similarity of their spatial patterns between a pair of features. Here is a set of simulated data of two variables. And here is their spatially smoothed data. The package I'm using is called GOR, which is analyzing the data by using a Gaussian process model to describe the distribution of the data. Here it uses a likelihood, maximal likelihood function to find the parameters that achieve the highest likelihood of the Gaussian process distribution. Compared with other packages, these packages could allow us to do model comparison between the spatial model and non-spatial model. Because this package is using a likelihood, it could calculate the maximal likelihood with assumption of spatial dependency and versus a likelihood without assuming any spatial dependencies. Then comparing these two likelihoods would allow us to conclude whether the spatial correlations is mostly determined by the marginal correlations, or the spatial correlations spatial models can better describe the data, the correlation structure between the two variables.
Quick question.
Mm-hmm.
So I'm familiar with AIC. What is BIC for model comparison?
Yeah, I still remember like there are both a linear is a log likelihood plus a certain constant, and the constant is actually depends on the number of parameters, and I think the constant is defined by like the number of parameters multiplied by another constant. I think it's just a constant difference, I think.
Oh, okay. Thank you.
Yeah, no problem. Thanks for the question. Yeah, here are a few packages listed here that could use to calculate the spatial correlations. Okay. So far, we have covered the spatial dependencies as well as the spatial correlations. Next, let's move on to the next problems, which is to extract major patterns of the features across the space. This is still an ongoing, a popular field in the computational biology, mostly because there's new sequencing technology developed in the recent few years. This technology is called spatial transcriptomics technology. This technology is allowing us to capture the gene expression within the context of the spatial locations. Here is how the data is generated. Firstly, a slice of tissue is cut at a certain position, and then a small slice is collected like this. And this slice could cover a few different layers or different anatomical regions in the tissue. After collecting this slice, this tissue slice will be put into a surface where there will be evenly distributed spots in the surface. And for the cells that are covered within each of the spots, their RNAs will be labeled uniquely. And then those cells, all the RNAs, will be sequenced, and the data will be generated in this format. Here is for each spot, the location of the spot will be provided, and the overall gene expression level of each of the genes will be also provided. Here, the plot is the gene expression of one of the genes in this data set. This technology is also allowing us to measure the gene expression level of different slices at the same time, where the slice could come from either the same positions or different positions. Here, the slides are cutting from the same mouse sagittal region, and here is the anterior half, and here is the posterior half. And each of the samples will be used to generate a separate spatial data. The spatial spatial expression data is useful given that the spatial structures of most of the tissues are very complex. For example, this plot shows the mouse sagittal section anatomical regions. And we can see that there are different, the anatomical regions have, each of the anatomical regions has own specific functions. And knowing the gene expression patterns of each of the anatomical regions will be able to provide us insights on the biological process within each region and how it's associated with its function. Using this data set, we can also study the interactions between different regions or between different cells within the same anatomical regions. One of the problems in analyzing this type of data is that in many data sets, the anatomical regions are not annotated in the data. And one step approaching to solve this problem is to identify the major variant spatial features of all the genes. Here is a method called multi-sample non-negative spatial factorization, in short as NMF, that developed by myself together with my supervisor, Dr. Casper Hansen, which could extract the spatial patterns from the multi-sample high-dimensional spatial data. Here is the multi-sample spatial transcriptomics data. Using our method, we could extract the spatial patterns that are highly associated with the anatomical regions in the tissue we are studying. Here are the factors that we could get using our method. We know that for some of the spatial patterns, we only see the spatial variance in one sample, but in another, for example, this is indicating some anatomical regions that is only existing in one half of the sagittal section. Here is the posterior section, but it's not in the other half. Here, the factors is denoting the cerebellum region. We can furtherly apply our approach to other multi-sample data, such as the data sets are composed of different genders or different disease conditions in order to find such spatial patterns that are sample specific, that could potentially reveal the biological process that are gender specific or disease specific. Our method has been examined using the multi-sample spatial transcriptomics data. However, our method can also be applied to more general high-dimensional spatial data with multi-samples. For example, we can apply our method to air pollution data in different countries to see if there are some shared patterns of air pollutions across different countries. Okay, I think that's the analysis tools that I want to discuss with you guys today. Maybe I would. So yeah, I'm not going through too much details of all those methods, but if you're interested, feel free to contact me using either Twitter or email. At last, I would like to thank my supervisor, Dr. Casper Hansen, and everyone in the lab, as well as everyone in the Stephanie Hicks lab. Thanks our Ladies Baltimore for organizing these events. It's such a great platform to communicate our knowledges in the data analysis. And thanks Alisa for the help with making the slides for the presentation. Okay. Yeah, that should be all. Any questions?
Thank you. Thank you, Yi. If people have any questions, feel free to drop them in the chat. I think Margaret can ask them for you, or like people are also allowed to unmute them, right?
Yeah.
Thank you.
I could add also, we'll post the slides if the Yi's okay with that. And we also recorded this session again. Yi, if you're okay with posting it, we can do that as well too. So if people have things that they want to review again, that'll be available on our, I guess, GitHub page for the slides and our YouTube channel for the recording.
Thank you for that presentation. I have a question concerning the data smoothing. I was wondering what interpolation method was used for that.
Something. Oh, you mean the visualization, the data smoothing here?
Yeah.
Yeah, I think there are many ways. Like I'm not sure if you are familiar with the traditional data smoothing, like smooth of a curve, like the Loess and the like spline curve smoothing.
Okay.
Yeah, they could be both like parametric and non like similar. There are similar ways. They could be a similar ways as and simple extension of like one-dimensional data smoothing.
Yeah.
And there are also, I think there are other approaches, but I think it's quite complicated that I firstly need to like here. Yeah, firstly, it will need to like fit a likelihood function to have an estimate of the parameters as a Gaussian process, which is like a multi-variate normal distribution and within a certain correlation structure. And then they will have a likelihood function of the data distribution. And then they also have like the observed data. And then they somehow combine those two data sets to have like posterior distribution.
Got it. Okay. So like I'm doing a project with spatial interpolation of, points everywhere. Is that different than data smoothing then?
Interpolation, I guess you mean you are, what do you mean by like interpolation?
I'm not really sure.
You mean like missing data, like you have some places where you don't have observations, but you're trying to sort of estimate what it would be there?
Yes.
Okay. I think there are also different ways to do this inference. Like you can simply just average around its, like for a certain location, you can just do average around its neighbors. Is that what you are doing?
Uh-huh.
Yeah.
Basically, yeah.
Yeah, that's a kind of like non-parametric ways, and there are also like parametric ways, say like you are kind of assuming like a more flat like surface across the neighbors, but the neighbors could be not like a flat surface. They could be like a convex surface or something. But yeah, if as long as your data point is dense enough and like they are very densely distributed in the two-dimensional space, that should be informative enough for you to do this interpolation.
Okay, thank you.
No problem.
You had a question in the chat. First, this is from Madeline Bauer that complimented you on your presentation And they had a question about the choice of parameters in this example. So how did you choose the parameters in the GOR example?
This one? Yeah, I think yeah, it's a great question. And here I think for it's also generally true for many packages. I think the developers have some suggestions on the parameters that they have tried different parameters, and empirically those parameters perform the best in their analysis. And here I think there are just those parameters are as an sense of the prior distributions. So right, I don't think those are another fact that some parameters should be changed dependent on the scope of the data. Like some data have larger scope, and some data are very densely distributed data points that their average distance between the points are very small, and some are very sparsely distributed point. In that case, some of the parameters will need to be adjusted based on the features of data. But for those, I think they are just empirically picked. And yeah, this is kind of which of the relationship of the distance and the correlation structures will you pick. And as my experience that it also depends on the data, and also depends on the like different type of data we are using. And in the spatial data, we are usually use the matterning, which is a similar of this exponential approach. But yeah, usually I think from developer side, they will just try different parameter, and they will have recommendation that if we are analyzing this type of data, we from our experience shows that these parameters will give you the best performance of modeling your data.
I'll just pass along another comment from the chat that someone really liked the slide on the different regions of the brain and enjoyed the use of spatial data analysis there. And Madeline said, thank you for your response to her question.
No problem. Yeah, I think the parameters of those could be tricky, like those Gaussian process. There are too many parameters in the GMOR as they are using a Bayesian modeling. So this could actually be a tricky problems. And also for the previous this method, there are also some variations of this method that using nearest neighbors to some method to define the neighbors to somehow speed up the computation, and the neighbors definition could also be tricky.
Do we have any other questions? Any other questions or comments? If not, thank you so much, Yi. This was a great presentation. And do you want to share the slide again where you've shared your contact details for people to reach out to?
Now or?
Oh yeah, right now, or just post it on in the chat.
Okay, I'll post
anything works. Awesome. Again, like I said before, we have a bunch of talks lined up in the future. Feel free to follow us on Twitter or our Slack channel. I think, should I share the slides again in case people are interested? People see the slide? so we have a new webpage here that will take you to events, which is just a meetup page for all our upcoming events in case people are interested in following. But yeah, other than that, thank you all for joining, and I hope you all have a wonderful weekend. We'll post the recording and the slides as well